Hey everyone, Dr. D here, and in this video, Tig and I are going to show you a nice overview of the material for Exam 3 in Biology 2420. So let's go ahead and get started. Okay, so here we have the slides. Exam 3 is going to begin here with Chapter 11. So let's get started. I'm just going to focus on, I know we've reviewed this in class, but I'm just going to focus on the important material for the exam. All right. One thing to know about archaea is that they live where it's not hospitable for humans. They live in anaerobic environments, salty environments, pH extremes, high temperature environments, Arctic temperature. And what you need to know is that there are two classes of archaea, the Euryarchaeota and the Crenarchaeota. Okay, very important that we understand Euryarchaeota and Crenarchaeota. The main difference is, if you look at this table, the Crenarchaeota are quite limited. They are the extremely thermophilic sulfur metabolizers. However, the Euryarchaeota are quite a bit more diverse. You see, they can be sulfur metabolizers, they can have cell walls, they can be cell wall-less, they could be extremely halophilic, they could be sulfur reducers, they could be methanogenic, so just be aware of that. The Euryarchaeota are quite a bit more diverse than the Crenarchaeota. Now moving on to bacteria, the dinococci and the non-proteobacteria gram-negatives. What do we need to know about these? Well, one thing I want to know for the exam is that Aquaficiae, the phylum Aquaficiae, is the oldest branch of bacteria. I want to know that the phylum Thermatogae is the second deepest branch, the second oldest branch of bacteria. And I want to know that Deinococcus thermus is resistant to desiccation and radiation. For Chlorobi, phylum Chlorobi, I just want to know that they are green sulfur bacteria. Phylum Chloroflexi, I want to know that they are green non-sulfur bacteria. For Cyanobacteria, I want to know that these are photosynthetic and responsible for much of the carbon fixation on the planet. For the phylum Chlamydiae, I want to know that they are intracellular parasites, which include chlamydia, which causes the disease chlamydia. You need to understand that these are obligate intracellular parasites. They cause chlamydia. For the phylum Spirochaetes, I want to know that they are spiral-shaped or helical-shaped because of their axial filament. And I want to know that they cause certain diseases like Lyme disease and syphilis. Okay, those are the things I want to know about those. Moving on to proteobacteria. The proteobacteria are also gram-negative bacteria. And the phylum consists of beta, gamma, alpha, epsilon, and delta proteobacteria. Okay, what do I want to know about the alpha proteobacteria? All I want to know about them is that they are oligotropic. I also want to know that, let's see, you can skip that. Rickettsia, I want to know about Rickettsia. What do I want to know about Rickettsia? This is what you should know about Rickettsia. You should know that they are parasitic or mutualistic, and they include Rickettsia prowazekil, Rickettsia typhi, Rickettsia rickettsii. Okay, the main one I want you to know about is Rickettsia rickettsii. This is the main one I want you to know about. This one causes Rocky Mountain spotted fever through tick bites. Okay, so just know Rickettsia parasite causes Rocky Mountain spotted fever. That's pretty interesting. What else do I want to know? I want to know that the Colobacteriae and the Hypomicrobacteriaceae, these form prosteca, okay, or a stalk, and they reproduce by budding. That's what I want to know about those. Moving on, I want to know that the Neisseria include the human pathogens Neisseria gonorrhoeae, which causes the disease gonorrhea. I want to know that Neisseria gonorrhoeae causes gonorrhea. I would definitely want to know that for the exam. What else do I want to know? I want to know that the methallotrophs are carbon users, one carbon users, so they use things like methane. They use methane as an energy source. I take. I want to know that the Pseudomonas are important for mineralization, and I want to know that the Vibrios are pathogens, such as Vibrio cholerae is a pathogen that causes cholera. Oh, all right. You going, Tig? I think Tig's on his way out. Bye. All right. I guess you guys can see me. Welcome. So I want to know, again, I want to know that Vibrio cholerae causes cholera. I want to know that Salmonella causes typhoid fever. Shigella causes dysentery. Klebsiella causes pneumonia. Yersinia causes the plague. Okay. And of course, let's know about E. coli. It's the best studied bacterium. Okay. What do I want to know about Delovibrio? I want to know that Delovibrio behave much like viruses in that they infect fellow bacterium. Once inside, they grow to quite a long size. Then they multiply by binary fission and then lyse the cell upon release. I want to know that Delovibrio can do this. Okay. What else do I want to know? I want to know about Helicobacter, specifically Helicobacter pylori. Why do I care about that? Because it causes peptic ulcer disease. That's what I want to know about those. Okay, moving on to the low GC gram-positive bacteria, the Firmicutes. I want to know they include the Mollicules, the Clostridia, the Bacilli. Let's go through these. The Mycoplasmas, or the Mollicules, what do you need to know about these? You need to know that the Mollicules are pleomorphic because they do not have a cell wall. And you might want to know that Mycoplasma pneumoniae causes pneumonia. With regard to Clostridia, what do I want to know about the genus Clostridium? I want to know that these are endospore formers. This is quite important to understand. Only two genus of bacteria can form endospores. Remember, Clostridium and Bacillus. You should know that. I want to know that Clostridium botulinum causes food poisoning or botulism. And I want to know that Clostridium tetani causes tetanus. Moving on to Bacillus, remember these are endospore forming and antibiotic producers. So remember what Dr. D says. What Dr. D says, if he could be any kind of bacteria, he would be an endospore forming, antibiotic producing bacillus. I would be a bacillus myself. If I could choose to come back as a bacteria, I would do that. What else? Staphylococcus, irregular clusters on the skin. So epidermidis is skin resident, aureus also a skin resident and also causes skin pathogens. That's all I want to know about those. MRSA and VRSA are strains of S. aureus that are particularly resistant to antibiotics. When we talk about lactobacillus, I just want to know that they are fermenters of lots of different dairy products, and they are also found in our normal flora on our bodies. Okay, these are the fermentation products. You don't need to know all the fermentation products. Next, the high GC gram-positive bacteria. They are actinobacteria. What about those? I want to know that the actinomycetes are a huge source of antibiotics. I want to know that they grow with hyphal development, so they're almost like fungi because they form a mycelium and they have hyphal cellular structure. They also have asexual spores. Not endospores. These are asexual spores for dissemination. Okay. What do I want to know about Corynebacterium? I want to know that Corynebacterium diphtheriae causes diphtheria. What do I want to know about Mycobacterium? I want to know that these are the bacteria with the waxy mycolic acids in their cell wall. And that's why these are called the acid-fast bacteria. And you should also know... Hey, Tig, you want back up? Wait, come on back. Welcome back, Tig. All right. Hey. Welcome back. Welcome back. All right. So what do I want to know about this? I want to know that Mycobacterium tuberculosis causes tuberculosis in humans. I want to know that No, don't. That's my drink. Thank you. He's trying to drink my drink. I want to know that Mycobacterium leprae causes leprosy. Okay. With regard to streptomyces, I want to know that they are responsible for that earthy smell after it rains because of the jasmine they produce. I want to know that they make up a lot of the soil microbiota, and they're a huge source of antibiotics. Moving on, the protists. I want to know about these supergroups. Okay, what do we need to know in particular? I might ask you which supergroup is known for possessing this cytosome, this feeding tube, or feeding groove, I should say, and those are the excavata. I want to know that Giardia causes diarrhea, and it's an example of fornicata. I want to know about Euglena, that it's photosynthetic. I want to know about trypanosomes. What do I want to know about that? They have this whip-like morphology. I want to know about Trypanosoma cruzi because it causes Chagas disease. It is spread by kissing bugs. And I want to know about Trypanosoma gambiense and Trypanosoma rhodiense because it causes African sleeping sickness, and it's spread by the vector tsetse fly. I want to know about amoeba. That's pretty self-explanatory. Some amoeba are naked, others have a shell. I want to know about amoeba proteus because that is your textbook amoeba. I want to know about the slime mold, specifically Myxogastria is the acellular slime mold, whereas Dictyostelium is the cellular slime mold. What else do I want to know? I want to know about Apicomplexans, like that Plasmodium has an apical complex, and it's an intracellular parasite. I want to know about dinoflagellates, how they cause the red blooms, right? They're marine plankton that causes those toxic blooms, and they can phosphoresce. Remember I showed you guys in class those blue tides, okay, red tides. Ciliophora, I want to know about paramecium. Paramecium has cilia. I want to know about diatoms. Oh, I might ask what group of protists has a heterocont flagella, and of course that would be the Straminopila. I want to know about diatoms, how they are photosynthetic and responsible for so much of the oxygen production on the planet. And one thing you should know for the exam is the difference between the Epitheca and the Hypotheca. Epitheca versus Hypotheca. Epitheca is the larger portion of the diatom shell. Hypotheca is the smaller part, okay, of the frustrule, I should say. Okay, then we talked about Archaeoplastida. You should know that these include the green algae, which is a group member here. On to the fungi. You should know that fungi are saprophytes. They are never photosynthetic. That means saprophytes means that they feed off dead organic material. Basically, they decay stuff. They could grow asexually or sexually. You should know all of these group members. These group members we talked about. Let's talk about them real quick. Let's see. Chytridiomycota. What do you need to know about the chytrids? That they are the simplest fungi. Okay, that's what you need to know about that. Zygomycota, you need to know that Rhizopus stolonifer or bread mold is an example. What do I want to know about Glomeromycota? I want to know that they are mycorrhizal symbiotes. They grow along with plant roots. Remember that? They help plants. Ascomycota, I want to know those are the sac fungi, and they include the yeasts, truffles, things like that. Basidiomycota, I want to know that these are your everyday mushrooms, toadstools, puffballs, etc. Microsporidia, I want to know that these are obligate intracellular parasites. And that about wraps up this packet. Let's move on to the next packet, which involves viruses. And let's talk about that. Read mode. Okay. Chapter 13, viruses. Let's talk about this. You should know the difference between viruses, viroids, virusoids, prions. We talked about all those. You should know what a virion is. It is a complete particle of a virus, the virion. You should know that all viruses have a nucleic acid genome, either DNA or RNA, enclosed by a coat of protein called the capsid. Okay. You should know about bacteriophage. You should know that some bacteria are enveloped, some are not, right? There are naked naked viruses that have no envelope. There are enveloped viruses as well. There's icosahedral shaped viruses, there's helical shaped viruses, there's complex shapes, etc. There's different sizes as well. You should know that protomers are the basic subunits of the capsid. You should know that, again, there's helical capsids. You should know that on icosahedral capsids, this is one tricky thing you should know. Icosahedral capsids, you should know that they have pentamers, and these pentamers are found at the vertices of the virus, and they have 5 subunits, whereas the hexamers are found at the edges, and those are 6 subunit capsimers. That's a tricky thing that you should know. Okay, there's also viruses of complex symmetry. You should know that the envelope can be taut or it can be loose. You should know what spike proteins are. Remember, hemagglutinin and neuraminidase are typical spike proteins, and those are found on flu virus. What else? You should know that the genome can be segmented or circular. There can be one piece or many. All right, we should know the steps of viral multiplication, that first the virus has to attach, and the specific tissue on which the virus can attach is the virus's tropism. We should know about the Berlin patient. How did treating the Berlin patients with bone marrow from a CCR5 delta 32 mutant donor, how did that treat the Berlin patient for both his lymphoma and his HIV infection? How did that prevent AIDS? So you should understand how that worked and understand what that had to do with absorption or attachment. Okay. All right, sorry you guys. Tig decided to cancel the video, so now I'm going to pick up where we left off. He kind of hit the escape button or backspace or I don't know, F12. I don't know, he does stuff like that. Anyway, let's keep going. So with viral entry and uncoating, that's pretty self-explanatory. Obviously, the virus enters the cell after it attaches to the cell. And then you have synthesis stage. Okay, synthesis stage where you're producing more of a genome, you're producing more of the mRNA, and you're producing more of the proteins as well. And then, of course, you have to assemble new virion and release, right? Remember that enveloped viruses tend to release via budding, whereas non-enveloped viruses usually lyse the cell upon release. You should know what temperate phage are, remember, versus virulent phage. You should understand lysogeny. Basically, lysogeny is an option for temperate phage. When a virus infects the host, that virus has 2 options. It can either enter the lytic cycle, which, don't cancel the video again, Tig. It can either enter the lytic cycle where the virus takes over the cell, reproduces, and then gets out of the cell, lyses the cell, or it could enter the lysogenic cycle, where the DNA gets incorporated into the host's genome as a prophage, then lies dormant. The bacteria, every time it divides, not only will it copy its own chromosome, it will copy the prophage DNA, the viral DNA, upon exposure to stress, such as UV. This will trigger excision of the prophage DNA and then entering the lytic cycle. So temperate phage have 2 options, lysogenic and lytic life cycles. What else? Remember, in eukaryotic cells, you can result in cancers. It can result in chronic infection, latent infection, acute infections as well. What else did we want to know? We wanted to know about viroids. You should know viroids are infectious agents entirely composed of circular single-stranded RNA. And these do not encode gene products. These cause plant diseases, and they work by triggering RNA silencing. So do know those facts. Virusoids, on the other hand, they do code for gene products, so they make genes. They have genes, and those genes code for proteins. An example is hepatitis D. Hepatitis D is not really a virus. It is a virusoid. It piggybacks in the capsid of things like hepatitis B virus in order to spread. Prions, we talked about these prion diseases. What you should know about prions is that it's a protein found in all of our brains, and in the normal folded state, it's not dangerous. However, the misfolded state is dangerous, and if you you eat brain matter or you eat meat from a diseased animal, that will introduce this misfolded protein into your system, and it will cause your normal proteins to misfold. And that mechanism is outlined here. What do we know? What else do we need to know about viruses? We need to know about remember these 7 groups here? We need to understand group 1, 2, 3, 4, 5, 6, 7. Please know group 1 is the double-stranded DNA genome. Group 2 is the single-stranded DNA genome. Know that group 3 is the double-stranded RNA genome. Group 4 is the plus-strand RNA. Group 5 is the negative-strand RNA. Group 6 is the retroviruses, and group 7 are the gapped viruses. Do you know that, please? So with group 1, what do we need to know about that? Simply that they have an easy time inside of your cells. The double-strand DNA uses your machinery to produce more RNA, more of its genome, and then assemble. Group 2, all you need to know about that is that it's basically like group 1 here, but except first you have to use a bacterial DNA polymerase to make double-strand DNA, and then you can take over the host machinery. With group 3, what do we need to know about this? We need to know that because this is an RNA genome, we have to make more RNA genome, right? So how do we do that? First of all, what you need to know about this, you need to know 2 major things. One is that the plus strand can be used as mRNA, so the ribosome can make all the proteins you need from that mRNA, including the enzyme required to copy. That's the second thing you need to know. The enzyme to copy the RNA genome to RNA to more RNA genome. That's called RNA-dependent RNA polymerase, and that can be coded for right here on this plus strand of RNA. So what you need to know is that these viruses do not necessarily need to bring with them the RNA-dependent RNA polymerase. They could code for it inside of the cell. These as well, group 4 as well. It's a plus strand RNA. It needs an RNA-dependent RNA polymerase in order to copy its genome. It can code for it in this plus strand RNA itself. Okay, it doesn't necessarily need to bring it with it. This strand can be used to make proteins. The proteins then package to form viruses. The negative strand viruses, these ones have it tougher. What you need to know is that these must, you see why I put underlined must bring? That's what you need to know. These must bring RNA-dependent RNA polymerase into the cell. Okay, they cannot code for it once inside because the ribosome cannot recognize negative strand RNA. It does not read it as mRNA. Next group, the retroviruses. What do we need to know about this? We need to know that these are RNA viruses. However, the first thing that's done inside of the cell is to convert the RNA to double-strand DNA, and the enzyme responsible for that is called a reverse transcriptase. It's essentially an RNA-dependent DNA polymerase, and what you need to know is that the virus needs to bring RNA-dependent DNA polymerase, aka reverse transcriptase, into the cell. What else do we need to know? We need to know about group 7. What you need to know about group 7 is that it's a double-strand DNA, but it's gapped. Look at this. The purple strand has a gap, and the blue strand has a nick. And it's a very strange-looking virus. But what you need to know about these ones is that to copy the genome, a pre-genome is required. You must form an RNA pre-genome. That's the special thing to understand about these ones. And that about leads me to the end of this review. I hope you guys have been studying hard. It's only those 2 PDF files that are going to be covered on this exam. And the things I mentioned are the most meaty subjects for the exam, where you can focus on those topics. And I hope it all helped And on behalf of myself and Tick, have a nice weekend, study hard, and we'll catch you next time.